This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 345, recorded on July 9th, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
How are you?
Very well, thanks. And yourself? You have to ask. Come on.
I know, we just did this for TWiP, didn't we?
We did.
And the weather here is pretty miserable.
Yeah, it's getting a little better out there. I mean, I can at least see New Jersey now. But
it's 21 Celsius, 80% humidity.
Yeah.
It's been raining all morning, right?
Yeah, it's dreary.
Rather muggy here in New York City.
That's right.
Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
I think you have the weather that we had overnight. It just poured rain all night, yeah.
Oh, dear.
That cleared up out there now?
Well, it's not raining now, and it's supposed to continue to clear up. But it's 63, which is 17 Celsius. A little cooler.
And you're getting ready to go up to Canada for ASV?
Yep, we'll drive up tomorrow. So.
Yeah, it's not a far drive.
How long a drive is that?
A little over 3 hours.
That's not
that's
bad.
Good. It'll be fun. Mm-hmm. And you have a Porsche with an 8-cylinder engine, right?
Not.
Do you have an electric car?
No, I have a Toyota, though. It's pretty fuel efficient.
There you go.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
You have an electric car?
No, I've actually thought about it. Honda now makes. We have a Honda Fit that my wife drives, and they now make an electric Fit,
Right.
Which looks like a really neat little car, but they so far they kind of charge a price premium for that.
Yep.
So not quite. It's the same reason we didn't get a hybrid, because it costs far more than it should for the car.
How's the weather up there?
It's crummy but improving.
Right.
So we've got broken to scattered clouds, medium height, and flash flood watch tonight, apparently. But tomorrow it's supposed to get better, and the weekend's supposed to be nice.
Right.
Yeah, well, Kathy and I will be in Western Ontario, London, Ontario, at Western University for ASV, which starts Saturday. And you should go to asv.org and check out the brand new website that I built. It's on. It's live.
And you can follow ASV 2015 on Twitter. It's at ASV 2015, and the hashtag is ASV 2015.
I went to, I guess it was the ASV meeting in London, Ontario.
Many years ago, yeah.
95, 96, somewhere in there.
Yeah, I was at that one also. So
Same place. They just changed the name of the university.
We're going back.
Right.
What was it then?
University of Western Ontario.
Yeah, it's Western University.
And where is Waterloo University? University of Waterloo? It's in that area somewhere,
It's
isn't
in
it?
Napoleon.
No.
It's in Waterloo, Ontario.
It's in Waterloo, Ontario, right? Which isn't far away.
But the funny thing is, and I had a conversation with a friend of mine from Edmonton, that it's now called Western Ontario, and it's kind of like the Ontario-centric view of Canada, because it's not very far west compared to Edmonton and BC.
The rest of Canada, yeah.
It's not called London, Ontario.
The university is what I'm talking about.
Those are different provinces,
Western
so
University.
they can still call themselves Western Ontario.
Okay.
Right.
This episode is brought to you by ASM, the Education Department. They have a cool workshop coming up called Turning Your Science into a Company.
Ooh.
October 1st through 3rd. You have to apply to get in. asmgap.org/TSC. Application deadline is August 20th. So if you've got some ideas and you're a scientist and you want to make a company out of it, this is a workshop for you. They're going to teach you how to do it. They're going to have people there who have done it, successful entrepreneurs. They'll give you tips on how to translate your science into a company. You're going to get practical tips, advice, resources on beginning your startup company. So check it out, asmgap.org/TSC. Application deadline August 20th. The meeting is October 1 to 3. I also want to tell you about a job, a virology tenure track position at the University of Toledo in the Department of Medical Microbiology and Immunology.
It's my old stomping ground.
Really?
Yeah, I was a professor there for 1 year.
Really? Out of your
with the
postdoc?
Zwickis. With the Zwickis.
Yeah, that's right. Well, they're still there.
How about that?
And in fact, the website used to be hosted at the University of Toledo, the ASV website, and so we just changed it. So we'll post a link to that, but if you are in the market for a job and you do the sorts of things they're interested in, microbiology and immunology, and of course virology is what they're looking for, check it out and look at the requirements. All right.
At least 2 people from that department will be at ASV. And there's also an event for department chairs or representatives who are hiring to meet with people in the tent. And I'm trying to pull it up in my email, but it's going to be listed in the special events in your meeting app. So if you're going to ASV and you're in the job market, you can meet with a variety of people who are going to be hiring in the next year.
All right. We have a follow-up, 2 follows up. And Kathy, can you take that first one from Steen?
From Steen, sure. Steen writes, thanks again for the recent focus on plant viruses, including our paper on TWiV 343. I think you all did an admirable job summarizing our results. Thanks especially to Kathy for feedback on the writing. Parentheses, note, my advisor wrote the paragraph you didn't like.
Love it.
Okay. I also enjoyed the recent florendovirus episode and Katie's follow-up on multipartite genomes of seed-transmitted Nipah viruses. And then he has a series of comments. The first one is on informative hosts. Regarding the question of using a natural host, why not try turnips? It is worth noting that tumor mosaic virus can be detected in...
Turnip.
What, isn't that what I said?
No, tumor. You said tumor.
Sorry. Yeah. I was thinking about, is it really mosaic? So I said the wrong thing. Sorry. This virus can be detected in wild Arabidopsis thaliana populations in Spain. Many plant viruses have a broad host range, and their names reflect the agricultural context in which they were discovered. I agree with the broader point that comparative genetic analysis of host virus specificity can be interesting and informative. Hopefully, CRISPR technology will expedite genetic analysis in turnips, yams, or whatever species the funding agencies prefer.
You should always put your turnips in the CRISPR.
Oh.
Alan.
Yeah. Big sigh. Okay.
Continue.
As Dickson mentioned, flowering plants appeared only about 130 million years ago, so we think many of the insights from Arabidopsis will translate. Hopefully, mouse-eared cress does not lie about the important things.
Wonderful.
So,
love it.
That was cute. Yeah. So then he talks about rub inoculation. Regarding Rich's question about cross-contamination, I do not think that we have ever detected virus coat protein in the mock-inoculated plants. We are more tender than the guy is in the wheat video. You have to gently caress the Arabidopsis thaliana leaves to avoid causing too much mechanical damage. Also, we do not really rinse off the plants afterward, especially not with a hose. Turnip mosaic virus is nice. The virions are not so stable on surfaces. Turnip crinkle virus, also used with Arabidopsis thaliana, reportedly requires much more care in avoiding cross-contamination. And the historically important tobacco mosaic virus is highly infectious on a large number of species. It's a pain. I just wanted to stop there for 2 comments. Fellow graduate student at UCSD with me worked on turnip crinkle virus, but she always called it killer crinkle virus. So, and then the story about tobacco mosaic virus being highly infectious. It wasn't there something about somebody smoking and cross-contaminating? Yes. I can't remember who or the story.
I met somebody at a meeting once who worked on TMV, and he said it was kind of ironic. They had this greenhouse full of tobacco plants and a huge sign saying "No smoking."
Yep. So I visited a vertical farm in College Station, Texas, associated with Texas A&M. At least they were involved in the original planning. It's called Caliber, and they use an Australian-derived tobacco plant for their host plant to manufacture insulin and factor 9 and stuff like this. And there's it's a no smoking zone for about a mile.
Wow.
If you smoke, you can't even go near that place because you could kill off the entire building's worth of plants. So very strict.
Just found a paper about this tobacco mosaic virus in cigarettes and saliva of smokers.
Yeah. Yeah.
Well, yeah, you can find it there. It's pretty stable.
It is.
Okay, so moving on.
Yes.
The next comment is about suppressor transgenes. Dickson asked whether transgenic expression of HC-Pro implants is possible. Indeed it is, but this makes Arabidopsis thaliana sterile. Transient or inducible expression can be used to get around this problem. Yeast RNAi Argonaut conservation. You briefly mentioned that Schizosaccharomyces pombe, fission yeast, has an Argonaut gene. S. pombe has been a great model for studying RNA-directed DNA methylation through RNAi pathways. Interestingly, the frequently used budding yeast, Saccharomyces cerevisiae, has lost RNAi, perhaps to better accommodate a helpful virus that kills off competitors. RNAi may have been discovered earlier if it were present in Saccharomyces cerevisiae. A review I linked to in my message noted that Argonaut genes, quote, have been linked to disorders such as Fragile X syndrome and some forms of autoimmune disease, end quote. So there is some link to human disease. Finally, petunias came up, so I wanted to mention that flower color in transgenic petunias has also been important for understanding RNAi, and that petunias are highly susceptible to several viruses. They are used as sentinel or canary in the coal mine plants for that reason. And then he mentions that it's 81 degrees Fahrenheit, 27 Celsius in St. Louis. Dark, ominous clouds outside my window. It has been a rainy summer. My colleagues' drought experiments in the field have turned into submergence tolerance experiments.
Nice.
Yeah. So the other comment I wanted to make was about the transgenic petunias, because when I was at University of Georgia, Tom Garatz came and was a visiting professor, was on sabbatical, or somehow came many times and was in collaboration with both Rich Meager and Judy Stromer. And so one or both of those labs switched and started doing some work with petunia, and I probably had some transgenic petunia, GMO petunia plants growing in my yard as a result.
Cool.
I don't know. They were multicolored, and I got them from their lab.
Yeah,
So,
I'm sure.
yeah.
They're gone now, right?
Oh, yeah. Long gone.
All right, Alan, can you take Andy's?
Sure. Andy writes, Hi, TWiVonauts. I was disheartened after hearing about the recent deluge of potentially fraudulent data surrounding publications by Olivier Voinet. I'm a graduate student working in plant virology biotechnology and found Voinet's name on a paper I was currently referencing for a presentation. This was troubling to me because I was literally basing one of my upcoming experiments on the conclusions of the paper. In my opinion, this kind of thing is especially harmful because now I don't know if I can trust the conclusions of the paper or if I should continue with the experiment. It seems one of the unfortunate drawbacks of the digital age is the temptation to use Photoshop as an alternative to rigorous science. Looking at some of the images in question, these are not even good fakes. The gel ladens were blatantly copied and pasted or simply flipped upside down in hopes no one would notice. I can't help but wonder if Voinet was only caught because of how blatant the fabrication was. This raises the disconcerting question of how many better fakes have gone undetected. On a humorous note, I ended up searching for other papers with Olivier Voinet's name that I may have read previously. In the utmost irony, I came across the following paper with Olivier Voinet as the sole author. And this is a review article entitled, How to Become Your Own Worst Enemy.
Yes.
From Nature Immunology in 2013.
It's about... go ahead.
Go ahead, Kathy.
Well, I was just going to say it's about viral persistence, right?
Yes.
It doesn't really have to do with his own situation.
No, absolutely.
It doesn't, but
this predates that by quite a bit. But
but he did become his own worst enemy, right?
Yes, he did, apparently. It's up to 110 Fahrenheit here in Arizona, and while we don't ever get any snow, we just got the first dust storm of the summer this past week. Every time you guys complain about the rain or clouds, I think, come take some of our sun. I'll take the rain.
Yeah.
I guess it's all relative. Yeah, but it's a dry heat.
Yeah.
Exactly.
Thank you all so much for the insightful and amusing podcast. I've been listening since my virology professor, Brenda Hoag, told our class about the podcast a few years ago. Now my girlfriend and I wait eagerly each Sunday for the new episode to be posted.
By the way, they just had a flash flood warning out there, so they showed an actual flash flood in progress on the evening news last night.
We're in Arizona?
Near Phoenix. Yeah, it was incredible. You could see all the debris, the leading edge of the water going right down this dry riverbed. It's only about 1.5 feet deep. As it crosses roads, however, the current could be so strong that it washes the cars away, and you die from being flipped over a number of times. So they have warning, flash flood zone, and you don't see anything. It looks like the rest of the desert except for this little channel, and it's actually quite scary to see these big flood events coming out of the mountains that you didn't even hear, and the next thing you know, people are dying because they think they can drive across them.
My grandfather and one of his colleagues had that happen to them in Texas, and my grandfather barely survived, and the other guy, they found the body a couple mile downstream.
Goodness gracious!
A couple of days later.
Yeah, I know when I go out west, they always say, "You see these dry riverbeds? Don't play in them."
That's right.
Yeah,
you never know when.
No, you drive across those fast.
Sure.
Yeah.
Wow.
Dear.
Needless to say, young scientists listening and old, don't fake data, please.
No.
It is totally against what we should be doing for science. And if you must fake data because you can't get yours to work, you need to get out of science.
Yeah.
And it doesn't help anyone. I just cannot understand how anyone could do this.
Ultimately, they get caught because people try to replicate the results and they can't. Then they check the results and the methods and they find out the whole thing's a sham.
I think a lot of them get caught, but not everyone.
Yeah, well, that's true.
Because I think some data nobody cares about.
Yeah,
no one
that's
checks,
right.
Which is fine, but,
Well, it's not fine, but it's...
It's not fine. It's fine in the sense that no one... this costs a lot of money and time when you have to sort this out.
No question.
It's not fine ever in any case, even if your data isn't important. But you shouldn't cheat. And I would think that you would have such pride in your work that you would never do this. I have always been totally puzzled at why people do this. Because I would never do it. I would get out of science before cheating.
Well, this comes from. I think this comes from a confusion of what you're supposed to be doing, because obviously when you start faking your data, you're not doing science anymore.
No, exactly.
But I think some people get so wrapped up in the other cruft around science, the career stuff and the advancement and the prestige and this and that they mistake that for the science.
Yeah.
And I need the prestige publication, and so I'm going to do whatever I can to get it, and that's just completely the wrong way to think about the whole process. You don't need the prestige publication. If you're in science, you should be there to do science and see what happens from there. But I just wanted to comment on this the two notions that this is A, becoming more common, and B, difficult to detect. I don't think it's becoming more common. I think we know there's been scientific fraud for centuries. Oh, yeah. It's just any human activity, you will get some people who will cheat. And there are some things that make it easier in the digital age, but at the same time, the tools for catching it have also become more automated. Mm-hmm. And I know I used to work, I did some work for the Journal of Cell Biology when Mike Rossner was there managing it, and he was very, very adept at and developed quite a few tools for detecting faked imagery. Because in cell biology, this is obviously a huge concern. Mm-hmm. And he actually, in a few cases, managed to detect things that were very sophisticated fakes. And there are other folks like him who've worked on these sorts of digital detective work techniques. And it is, yes, of course, unfortunate that has to happen. But yeah, there are going to be people like this, and they will eventually get caught either by somebody analyzing the images or by somebody trying to advance the science and finding that the system doesn't work.
What's cruft?
Junk.
I just pasted it in from a Google search.
And one of a couple of theories for the origin of the term, but yes.
Yeah. And one of so it seems to be a computing term, badly designed, unnecessarily complicated or unwanted code or software anything that's left over, redundant, getting in the way.
Yeah.
Number one, the dust that gathers underneath a bed. Somebody's used it for.
Yeah.
Sure.
That's a good.
Those are dust bunnies. They already have a name.
Dust bunnies, yeah.
Cruft. That's a
Already.
good word, cruft.
Yeah.
All right. We have a couple of snippets and a paper. First snippet comes to us courtesy of Dickson Despommier.
Indeed.
Which is good because I was thinking this is right up his alley.
And it is. It
And
is.
you mentioned it on last TWiV, is that right?
Yep.
So you sent it, and now let's snippet it. It's from the
did.
American Journal of Tropical Medicine and Hygiene.
Right.
From a group at Fort Collins, Colorado. Centers for
Yes.
Disease Control and Prevention, the National Center for Atmospheric Research.
Yep.
Actually, Boulder and Fort Collins.
Yeah.
Meteorological conditions associated with increased incidence of West Nile virus disease in the U.S. 2004 to 2012.
And by the way, this is probably the most TWiV paper I can imagine.
Yeah.
It is truly This Week in Meteorology and Virology.
That's you put them right
Yeah,
together.
it really is. It's true.
That's true. And we've talked about this in the past. In fact, the second episode of TWiV was, I believe, on the West Nile virus, if I recall. So Vincent advised me on this little book that I produced called West Nile Story. And in this book, I document through past epidemics of West Nile virus throughout Europe and Africa that they're always associated with droughty conditions followed by high temperatures and then a rain, which is, of course, typical for droughts followed by rains. And that's just what we were discussing just a moment ago, in fact. And it seems as though that pattern should hold true no matter where you find West Nile virus. So when it arrived on our shores in 1999, I began tracking the weather conditions with regards to the spread of the virus as it worked its way across the United States. And indeed, the patterns were very clear. And I had students, graduate students working for me that were also collecting data from meteorological studies and from the CDC to correlate those two things together. So every time the temperature went above 90 degrees and there was a drought for longer than 3 or 4 weeks, that was followed by a human case of West Nile virus. So this is a paper that actually follows up on that concept and details in much greater detail the actual meteorological conditions that favor the spread of the virus, not to birds, but rather to people.
Of course, this is a mosquito-borne disease.
Yep.
So looking at weather conditions is a way of looking at how they affect mosquitoes
Exactly.
and transmit the
But
virus.
it's a special mosquito, right? It's not just mosquitoes. These
All
are
mosquitoes are special.
that's right. But this mosquito thrives during droughts in the presence of
Culex
humans.
mosquitoes, right?
Yeah, because they love polluted water, and we're very good at creating polluted water.
And this mosquito takes the virus from a bird reservoir
Right.
and brings it to people.
Or it vertically transmits it to its offspring.
With mosquitoes, okay.
Yep.
So what they did here is they developed models to identify meteorological anomalies
Yeah,
associated with above-average incidence of West Nile virus neuroinvasive disease.
right.
So they have county-level
Yeah,
West Nile data and meteorological data,
that's right.
right?
That's right.
And they try and make correlations. And so what they conclude is that above annual temperature is associated with increased likelihood of higher than normal West Nile disease incidence
Exactly.
nationally and in most regions.
That's right.
Lower than average annual total precipitation was associated with higher disease in the eastern U.S., but the opposite was true in most western regions. Okay, so that's where the model breaks down, if you will, because there's
Yeah.
a difference between east and west. Right?
But I would argue also that there are 2 endemic centers for West Nile virus in the United States right now that never fails to exhibit the highest number of cases annually, and that is Texas and California. And the Imperial Valley of California is where you have all the migrant workers, and the same is true for Texas. And there's a lot of agriculture that goes on there. I will predict that because of this 4-year drought that California is suffering from, that the amount of irrigation has gone way down,
right.
and therefore the breeding sites for Culex have gone way down. And of course, the migrant workers have gone way down because there's nothing to harvest. So California could drop off the map in terms of incidence for West Nile this year.
People are always projecting California dropping off the map.
This is all true. There's no question about it.
Literally.
Yeah, but it's a great article because they produce maps, and the thing about this is that when my students were working on these, trying to gather more information from the states with regards to the counties that were affected and how many people and when did they become infected, that data was not usually given out. They said, I'm sorry, this is proprietary, and we are only obliged to report this to the Centers for Disease Control. So here's the Centers for Disease Control now finally massaging this data into a very, very readable and understandable scenario.
So they do a little speculation on how these meteorological anomalies could affect West Nile. They say higher temperature favors greater larval and adult mosquito abundance. It may also impact bird migration, hatching, or bird community composition, and may also modify human behavior and influence exposure to mosquitoes. I guess when it gets warm, people go outside more, right?
Right.
Go hiking around and so forth.
Right.
What about the rainfall? Okay, how do they explain the differences? So they say there are differences in the way water sits in the east versus the west. Yeah. So, for example, in the western areas, excess rainfall may increase risk by creating temporary standing pools of water for female mosquitoes. Yeah. Temporary standing pools.
Yep.
Right. The relationship between precipitation and disease in the northern Rockies and Plains mirrored that of wetter eastern regions was unexpected.
Yeah. How about that?
So I just recently covered a conference for the New York Academy of Sciences on climate change and health. And which I kind of thought could have been titled the "Another Reason We're So Screwed" conference. But so one of the interesting themes to come out of it is that with a lot of diseases, and especially with vector-borne diseases, it's all about the microclimate. So you can find mosquitoes in places that you wouldn't think of as mosquito habitat if the particular microenvironment of that little area, and by little area I mean it could be just one little ravine in the mountains. If it matches what the mosquito needs, then you get them there.
Yeah.
And it occurred to me in reading this paper, I think the apparent contradiction that drier in the east and wetter in the west gives you more West Nile. It may just be because of what the background climates are like in these different places. So you think of the Northeast up here in Massachusetts. When you say drier than average for Massachusetts, that would be wetter than average for West Texas.
Sure, but you also have to throw in temperature there, Alan.
Of course, yes. But even if you go to South Carolina, drier than average in South Carolina is going to be wetter than average in West Texas.
Yeah, that's true.
So I think they may be, since they're looking at regional averages in regions that have very different, mesoscale phenomena going on. That may be what's causing this apparent contradiction. But if you were to look at the actual microenvironments there, and I realize the problem with looking at that is even in a well-monitored place like the U.S., the data only gets so good.
You're right.
But if you could monitor down to the level of smaller than county level, you might be able to start getting at the exact microenvironmental conditions that are going to give you this sort of thing, and that's where you get to maybe even predicting outbreaks.
But that's the data that I wanted and I couldn't get because these counties have the location of each of those people that got infected.
Yes.
And that's not a data that you can get, so at least I couldn't get it.
Well, it's difficult to anonymize.
And you're darn right. I mean, unless it's an apartment house, if it's a single-family dwelling, you know exactly who it was.
Even if it's an apartment house. I mean, this is
you're right.
An ongoing problem with medical data. As soon as you geocode it and you've got something like a birth date in it,
you're right.
If you know a birth date and a zip code, you could probably identify one person.
Yeah,
that's right.
So they're that is probably a lot of the reluctance surrounding that. But if these can be made, if there's some way to make these data usable by people without necessarily exposing that information, that would be good. I don't know. Maybe the CDC can work on developing algorithms that people could access without exposing the data.
So the data that we gathered is listed on our medical ecology website under West Nile, and invariably we didn't have a single human case until the temperature hit 90 degrees during that drought. If it was lower than that, there were no human cases, and the explanation is kind of vague. And the Israelis did some early studies on West Nile back in the 50s and showed the viremia in the mosquitoes themselves, which was related to the ambient temperature.
Yeah.
So that if the temperature never exceeds 90 degrees, then the number of viral particles delivered per bite is low, and therefore the people are not susceptible to that dose, but birds still are. And that was the break point, and I think it's still probably valid.
Dickson, you will like this statement. In addition to temperature and precipitation, many other factors influence West Nile.
Yeah.
Among these are seasonal shifts in mosquito feeding preferences from amplifying bird hosts to humans after birds have migrated.
There you go.
So I know you used to say when the water goes, the birds go.
That's right.
And that may be part of the shift in preference.
So of another flavivirus, Eastern equine encephalitis is exactly like that, except that it's all due to bird migration, right? Because they bring it down, give it to the horses, and the horses give it to the mosquitoes, and the mosquitoes give it to us. Interesting.
So it's a pretty readable paper. Check it out.
Yep.
And as you said, they do, as Alan said, they do subscribe to this idea that you have to look regional, but Alan is saying even more than regional. You have to even look more locally, but that's sometimes difficult to do. It's pretty cool.
Right.
Our next snippet was suggested by a number of listeners. Gabriel Victora, who is a B-cell biologist who has been on TWiV. He sent this paper. Maybe you saw this. Not exactly virology, but probably argues that the cause is not viral. However, I would disagree with that.
I would disagree
conclusion, and we will see why in a moment. Tate writes, here's a paper on rituximab in ME/CFS. It was open label with the placebo group from the first study, but I think they gave maintenance doses. And also Ricardo sent a link to this paper. So this is a paper published in PLOS One. The title is B lymphocyte depletion in myalgic encephalopathy chronic fatigue syndrome, an open label phase 2 study with rituximab maintenance treatment. So this is chronic fatigue syndrome patients, which we've talked about many times here on TWiV. And this is a follow-up of a study that was done a couple of years ago where they treated patients with, I think, 1 or 2 doses of rituximab, which is a monoclonal antibody against a protein on B cells called CD20. And the effect of giving people this monoclonal is it destroys B cells by a number of mechanisms and reduces the number of B cells, and B cells make antibodies. And in that first trial, just with the limited number of doses, there was an improvement in some of the patients. So they wanted to do a new study where they gave more doses over a longer time of this rituximab. This rituximab, by the way, has been approved for use for other conditions. This is
a leukemia treatment.
Leukemia treatment. If you have too many B cells, you get this and kills them. So it's FDA approved, and it's been shown to be rather safe with minimal side effects. Although I think if you get rid of your B cells for a long time, this could be...
it basically has the side effects you would expect from wiping out your B cells.
So you can get opportunistic infections of various sorts, so you have to be careful. So I'm not sure you'd want to take this your entire life, but it points out that what might be going on in this disease. So what they did in this study is they did multiple dosing, and they also used some of the placebo patients from the previous study. So apparently when you do a follow-up, that's required that you offer the placebo group the opportunity to receive actually the drug, right?
Right. So that's what makes this an open label trial, because both the researchers and the participants know what treatment is being administered.
Right.
This is as opposed to a blinded trial.
A blinded trial, right. So they had 29 patients here with confirmed ME/CFS according to Fukuda and Canadian diagnostic criteria, right? Both Fukuda and Canadian, I believe. I know
think so.
For all of them. And the Canadian, I think, is more restrictive than the Fukuda criteria. So it's a number of criteria that you have to fulfill. And the way this was done is they gave a number of doses. Let's see. You initially got a dose, and then you got follow-up doses at 3, 6, 10, and 15 months. Is that correct? Hang on. Where is it here?
Sorry, I just noticed inclusion criteria were diagnosis of ME/CFS according to the Fukuda 1994 criteria only.
Right, and I think a number of them also satisfied the Canadian criteria as well. I remember,
right, but that wasn't
required for the study. So, what were when were the doses given? Do we remember? I can't.
Yeah,
So basically, they would give multiple doses, and then the patients would fill out a questionnaire and record a number of scores on different aspects of their well-being. For example, fatigue score, cognitive scores, pain, physical health, mental health, physical function, body pain, et cetera, et cetera. So there are a number of numbers they'd have to fill out. And then there was a goal of the trial was to achieve a certain score. All right.
And the infusions were at 3, 6, 10, and 15 months.
Yeah, right. And then the follow-up went out to 36 months.
Right. They followed up out to 2.5 years.
So this is the difference between this and the earlier study is that here we're giving multiple doses over an extended period of time. And so basically significant responses were seen in 18 out of the 28 patients, 64%. Okay? So they had marked improvements in many of these criteria, which is presumably attributable to suppressing the number of B cells, which they did measure over the course of this. And then after they stopped the trial, they found that their B cell numbers went back up again. And so 18 out of 28. So 10 patients still did not respond. So this is not apparently going to help all ME/CFS patients. So maybe it's a disease that has a variety of causes, but for some it looks like getting rid of B cells really helps. So why would that be? Well, of course, since they make antibodies, you could say that maybe. Well, one of my ideas was that you're making antibodies which are cross-reacting with your own tissues.
Yes,
right?
Which is another theme we're going to come to in the next discussion.
And so you suppress the production of those antibodies. Now, you should know that the improvement in symptoms has a lag after the first treatment. And they're saying this is consistent with, long-lived antibody being present. It takes a while for those levels to go down after you destroy the B cells. It could be that some of those are cross-reacting with your own tissues, and that causes the symptoms of this disease. Now, because a lot of ME/CFS patients report the onset of symptoms not too long after an infection of some kind, it could be that one or more virus infections is leading to an autoimmune response. So I would suggest that you do a study where you do viro scans of ME/CFS patients, and we talked about this a couple weeks ago. You look at all the antibodies against viruses that these patients, and see if there's a commonality, or maybe an epitope that you can discover. And we'll talk about this in the main paper here. That is really responsible for this, right?
Yeah.
Because they all have multiple viral infections, as we've discussed, of various sorts. And many other people have the same virus infections, but maybe in this particular group, for some reason, a particular epitope is cross-reacting with a cellular one.
And that would actually put this into a whole category of diseases that have that kind of an etiology. I mean, lupus is an autoimmune disease against DNA.
Right.
And type 1 diabetes.
Type 1 diabetes, I was just about to mention, is you get a viral infection, you get a cross-reaction, and you get, you end up with diabetes.
Indeed.
There's a genetic predisposition to that as well, but there may be genetic factors in this too.
Hear, hear.
So again, it's not all the patients. So maybe the others have a different etiology, but at least in a good fraction. So I would guess that it would be useful to do a bigger study, right, with more people and see if this continues. Where are these patients from? Let me see. I have a feeling because the study is in Norway.
Study's in Norway. I'm guessing the patients are in Norway.
Yeah, so I would, of course, like to see this done in other countries, right?
Yep. So if you had a twin that didn't have ME/CSF, could you do a whole bone marrow transplant into the one that did and restore them to normal function if it's an autoimmune disease?
So would that get rid of all the B cells that bone marrow?
Like they do for transplantations.
Yeah, but aren't they in lymph nodes as well? Is that going to get rid of those too? I guess total body irradiation would.
Right.
Yeah. I guess it could, but that's a rather dangerous thing to do.
Of course it is.
You'd have to do that ethically, you'd have to find somebody who was getting that done for some other reason. But then, of course, you wouldn't be able to diagnose them with ME/CFS because they would have
something
else.
I mean, it would be better to identify the particular B cells that are producing.
Well,
these
much better, of
antibodies,
course,
right?
of course. But, so the other question is, have they done twin studies on this to see whether or not there is a genetics to it or not?
I don't know.
I'm sure this has been done. I don't know this literature.
I don't either.
All that well. This is Norway. They're actually starting a new double-blinded, placebo-controlled phase 3 study at 5 places in Norway.
Good.
And that's been started already. 152 patients. That's great. I think they should do it in other countries as well.
I think of this as more. Obviously, what they're trying to do is they're hoping that this already approved drug will be useful as a treatment, and yes, that'd be great. But from what I get from this paper, what I think is actually the bigger, more useful thing is that this gives a lead on where the problem may be.
Yeah, exactly.
So this may not be the real way to treat this, but it suggests that B cells are the place to look for causes.
I found a blog post about twin study, and it says, shoot, now I lost it. Oh, rats. It was a proteome study, and I can't find the thing I was looking for. This was a twin study. Twin studies, they had, thus far, twin studies have had mixed results with ME/CFS. And this study involved just one set of twins, but an intriguing set of twins, similar lifestyles, backgrounds, locations, and working environments, but something only went wrong for one of them. So just answer the question. There have been twin studies for ME/CFS.
Okay, sure.
Also, another interesting comment made in the discussion, they say, an observation that may support an autoimmune mechanism is a moderate but significant increase in the risk of B-cell lymphomas in elderly CFS patients, indicating a chronically activated B-cell system. So I should say that, I'm suggesting that it's an antibody cross-reactive issue, but, there are other effects of rituximab.
Sure.
Off target, I guess, and they could participate as well. I noticed, Alan, they abbreviate autoimmune disease as AD, the Allen-Dove disease.
Great.
The presence of Allen-Dove in the general population was 3% in the U.S.
Oh gosh, we're all hosed now.
And 5% in Denmark. You got 5% Allen-Doves in Denmark.
Wow.
So it's all context dependent because that's also an abbreviation for Alzheimer's disease.
Yes.
It is, yeah. So among patients in the present study, 41% had one or more first-degree relatives with an autoimmune disease. And that's compared to the prevalence of AD in the general population, 3 to 5%, right? So it's overrepresented in these patients. And so that may be higher. That's higher than expected.
And I would actually argue the Denmark point is relevant because Scandinavian populations are often more genetically isolated than some other European populations, and certainly than, say, American populations. And that's another point we're going to come up to in the next paper when we get to it. That you may actually have a higher signal and less noise, and this may be a particularly good place to study that.
And I should also point out that other studies have suggested autoimmunity as a mechanism. This is not the first
yes.
one.
Oh, no, this isn't the new
but
idea.
the results of this study are consistent with it. So,
and other studies have suggested kind of generalized autoimmunity issues, but here you've got a somewhat specific answer on B cells.
So we'll see. Maybe it's some particular combination of viral infections or a particular epitope that's cross-reactive. We'll see. I think it's a good study. And that brings us to our paper here.
Oops.
Is that you, Dickson?
It certainly was.
All right.
Putting on my glasses.
This is published in Science Translational Medicine. And actually, Rich and Alan, who sent this out? Did you, Alan, to all of us?
I did, yeah.
And Alan said, we should probably cover this sometime. That's an Alan Doveism. We should probably...
Yeah, maybe we ought to do this. So this has been percolating along in the news for a while, and I get updates from the flu vaccine community on a regular basis.
We also have talked about it on TWiV, right?
Yeah, and we've talked about it on TWiV, this weird thing that happened with the 2009 H1N1, one particular pandemic vaccine, Pandemrix, which was associated with, apparently associated with a slight increase in narcolepsy risk in Finland. And it's been... the manufacturers have been very, very, Novartis makes Pandemrix or made Pandemrix, and they've been very aggressive about figuring out what the heck is going on here. But it has not been easy because we're talking about small numbers of cases, but it's kind of like the Guillain-Barré situation where there is a detectable difference, and you look at the statistics and you say, yeah, it looks like these people who got this shot do have a higher risk of narcolepsy.
Yeah. So the highest rates, this Pandemrix was given to 30 million people in the EU countries, and as of January this year, there were over 1,300 cases of vaccine-associated narcolepsy reported, whereas so this is an inactivated vaccine that includes an adjuvant called AS03, and in contrast, there's another vaccine called Phocitria, which is adjuvanted with a different adjuvant, NMF59. There have been 6.5 million doses in the EU and 25 million doses in Europe and Latin America. No increased risk for narcolepsy in those cases. Interestingly, in China, there has been an association of narcolepsy with actual infection with the 2009 H1N1 pandemic influenza virus. So this is saying that there's not just a vaccine involved, but something that's also present in the infectious virus. So there have been a lot of studies. We talked about this a couple of years ago. Been lots of epidemiological studies on the number of cases in different countries and what they're associated with. And this Pandemrix looks like it's clearly associated with it. So what is narcolepsy? Well, it's a chronic disorder where you have excessive daytime sleepiness. And it's due to loss of hypothalamic cells, and this causes a deficiency in a neuropeptide called hypocretin, which is abbreviated HCRT. And these patients have deficient hypocretin in cerebrospinal fluid. We don't know why, but in dogs, dogs with hereditary narcolepsy have a mutation in the receptor for hypocretin. There are also murine models for this disease with mutations in the receptor or in the gene encoding the neuropeptide, and these cause narcolepsy.
And just a more general description of narcolepsy. It typically begins in the teens and early 20s, but it can occur as early as 5 years of age or after 40. And there's two types, one with cataplexy and one without cataplexy.
What's cataplexy?
Yeah, somebody help me out here.
Okay, we'll look it up.
Yeah.
Cataplexy.
It's like
Yeah.
a catatonic kind
It's
of
a medical
trance.
condition in which strong
do
emotion
it earlier.
or
It's in the
laughter-
paper even. It's mentioned.
Sudden physical collapse.
Right.
That's right. Okay, yeah. And basically, patients will have falling asleep, just in the middle of the day. And so there are some treatments for it, scheduling daytime naps, avoiding drugs that produce daytime sleepiness or insomnia, and having a psychosocial support group. And then there are some pharmacological things that can be done also.
Shouldn't be operating heavy machinery, I presume,
Right.
or cars.
So
Yeah, this rules you out of certain jobs.
Yeah.
So we should note that the narcolepsy that was associated with vaccination, usually the onset was a couple of months to 10 to 14 months after immunization. All right, so
And particularly in pediatric patients.
so
Right.
in some countries that's because the pandemics was mainly given to kids.
And something different was given to adults?
Yeah, so for example, in the Netherlands, the other vaccine was given to older people.
Oh yeah, they have there's a high dose. They're doing this in the U.S. now too. There's a high dose flu vaccine for people over 65 in the U.S. and I think similar age in Europe.
Yeah. So in Netherlands, Pandemrix was offered to children 6 months to 5 years of age, whereas Focitria was offered to older children as part of a large national campaign. I don't know why that was done.
It's just a
dosage, you think?
Yeah, they formulate vaccines for a variety of different groups of people in the interest of trying to make it more effective for those groups, because the epidemiology suggests that you need to stimulate responses differently in these different groups.
So in the Netherlands, 654,000 people got Focitria, and no narcolepsy there, but 588,000 got Pandemrix. These were kids, 20 cases of narcolepsy. So
Interesting.
really associated with this one vaccine. So they said, what's the difference between these two vaccines? So they're slightly different. Flu vaccines are usually recombinants, reassortants with high-yielding, egg-yielding viruses, and they can be slightly different for these different companies. So they said, they looked through the sequences of the proteins in these two vaccines, Pandemrix and Focitria, and said, what is different among them? And they identified one hemagglutinin sequence fragment and two fragments from the nucleoprotein that had an amino acid difference between the two vaccines. So they're comparing, the two, which gave rise to narcolepsy and the other didn't. And they could find a couple of short amino acid sequences, which we call peptides, that differ, all right, HA and NP. So right away they said, maybe this is giving rise to an autoimmune reaction where you make antibodies against this particular peptide and it cross-reacts with something in your body. And they're looking at the receptor for hypocretin because that's what seems to be involved in narcolepsy. So then they say, is there any sequence similarity between these viral peptides and peptides from hypocretin? And the answer is yes.
That was a pretty cool step, I thought.
Sometimes good science starts with a lucky guess.
Yeah. So the mimic peptide from one of these NP has residues that are in common with a peptide from HCRT, hypocretin receptor. It's amazing, right?
Yeah.
If you compared every protein with every other protein in the world,
That's
you might get this.
Yeah,
but
that's right.
no one's doing that. So here they had something very specific, and they looked at it and they got it. And they said, okay.
Well, and what allows you to do this is that there's enough basic science on both the virus and on narcolepsy that you can say, well, narcolepsy, what causes that? Well, we're not exactly sure, but we know it involves this hypocretin and hypocretin receptor. Yeah. We've got the human genome sequence, and you've got all this stuff that you can mine. And then the flu vaccine, of course, you can profile the proteins like this and do this analysis and say, hey, these two things are a tantalizing clue.
So the NP, one of the NPs matched up, YDKEEIRRIWR, those are the single amino acid abbreviations. And that is similar to a part of HCRT receptor 2, YDDEEFLRYLWR. Right? So it's the one from X, what the vaccine is called X179, which is Pandemrix. So the Pandemrix closely matches, the Pandemrix NP peptide closely matches that of this HCRT receptor. So do people have antibodies to HCRT receptor, and does it have anything to do with being immunized with Pandemrix? So what they do here is they have human cell lines that produce the HCRT receptor, and they simply take sera from people who were immunized with Pandemrix, and they say, do they react with these cells? And the answer is yes.
Yes.
A significant proportion of these. This is day 500 post-vaccine. They got what they could. It would be nice if you could get a timed series of sera, but they react with. They have antibodies to their own HCRT receptor.
Wow. I could just imagine developing the results on that.
Oh yeah.
experiment.
Yeah.
Sure. Wow.
I mean, it's clearly HCRT because they do co-localization with a known antibody against the receptor. It's clearly there. Mm-hmm. These peptides that are in the NP, they compete with antibody binding to the HCRT receptor, so it all makes perfect sense. You get this vaccine, you make antibodies to this NP, and those also cross-react with HCRT. ELISA, they did immunofluorescence to confirm this again. Now, the interesting thing here is that the two vaccines have NP in this peptide that differs by one residue, but that doesn't influence the ability of the peptide to block the interaction of the antibody to the HCRT. So that doesn't seem to be the mechanism here. But what they did find is that the peptides bind MHC proteins differently. There happens to be an MHC allele that's associated with narcolepsy. Right. So MHC is a protein on the surface of many cells which presents antigens to immune cells. And there's a particular MHC called HLA DQB1 asterisk 0602. So this simply means it's a particular sequence of a MHC gene that in people with narcolepsy, that a higher than expected number have this sequence.
Wow.
Right. And they're thinking, what if this peptide is also differentially binding to this particular one?
Right.
And in fact, they do binding of the peptides and in fact to the MHC proteins. And this variant one that's in Pandemrix binds better to this variant MHC allele.
Right. What I wanted to say earlier, and it now comes around full circle, is that when you pointed out that the Pandemrix peptide had the amino acid similarity, a little bit closer. It was only one amino acid more similar than the other peptides. So, in fact, both peptides could do that antibody blocking that you said.
Right.
But when you get down to this binding to the MHC, the one with the particular amino acid sequence is potentially better.
Right. So the reason this is important is because the B cells are going to be presenting antigen to T helper cells, and the T helper cell, the T cell receptor, is going to look at the MHC molecule. It's going to see the peptides in it, and if it detects it as foreign, it's going to produce cytokines that enhance the ability of the B cells to make antibodies. So if this particular NP peptide fits really well into this MHC, then those B cells are going to be making lots of antibodies against that particular sequence, and that could influence the autoimmunity.
Amazing.
It's very cool.
But wait, there's more.
There has to be more.
So the next part is asking how much NP protein is present in these vaccines. So they have a whole bunch of different vaccines that are made in different ones. Besides Phocetria and Pandemrix, they have Fluzone and Fluarix and Afluria and Fluvirin, Agripple,
very creative names.
So they look by Western blotting and then by mass spectrometry, because for some of them they can't even detect the NP. And guess when you compare Pandemrix with Phocetria. Which do you think has the highest level of NP protein in it?
Pandemrix.
Pandemrix. And
How many guesses do I get?
Pandemrix has the highest amount of NP protein, and Phocetria has the lowest. So again, Pandemrix is associated with narcolepsy. Phocetria is not. And it seems to be correlated with NP
That's
protein.
quite remarkable.
And
So this is a vaccine that just happened to end up with a variant NP protein
right.
that happens to bind
Amazing.
with higher affinity to a particular HLA type that happens to be in people predisposed to narcolepsy, and the vaccine happens to have an especially high concentration of this nucleoprotein.
Right.
Yeah.
This is just an appalling string of bad luck.
I'll say.
Which you couldn't have predicted at all.
No, there's no way to see something like this coming. This is
But you've got hindsight now. So
this
you
is
can-
getting hit by a meteor while winning the lottery. I mean, it's
So you can go now and use this data, though, right, to survey who is prone to getting narcolepsy and then exclude them from
you can
this
do all
particular-
kinds of things
Yeah,
now that you
this
do
is great. This is really
So this we
good
should point out
public
this
health
is
intro.
this is quite rare. So this is really a rare adverse event,
It's
right?
true.
It's 1 in 16,000 to 1 in 50,000 doses. Do you see this? But you don't want this to happen, obviously.
No.
So you could either eliminate NP from
You could do
the
that.
vaccine.
But does it enhance protection against
That's
influenza?
That's a good question. I mean, both vaccines are protective to the certain. Phocetria and Pandemrix, as far as I know, are equally.
They both produce antibody responses.
So, that's a good question, whether you could take it out entirely and get around this.
Right.
So you can put in less nucleoprotein. You can make sure it's not this nucleoprotein. You can make sure you're not giving it to populations that have a lot of this particular HLA type.
Right.
There are all kinds of ways to make sure this never happens again. I mean, the odds of this ever happening again are slim to none anyway. But now you have a grip on exactly how to be certain. That
right now you don't use Pandemrix anymore, right?
Oh no, nobody's using that. I don't think.
Just use the other ones, but
Yeah.
they tried
In fact, that happened right as this was coming out. They basically Novartis paused everything, said, "Whoa, use other vaccines," and people did. And
So you now have identified the species of antibody that's responsible for inducing narcolepsy. You could use a RNAi approach to get rid of that antibody without any harm to anything else and restore these people to normal health. Potentially.
I just wanted to make a correction. Pandemrix was GlaxoSmithKline.
I'm sorry, GlaxoSmithKline, not Novartis.
Right.
Sorry, everybody at Novartis.
I know some people there, so. I don't know if this is going to come up, but one thing that when I was reading the abstract, just starting out, and they talked about the fact that these are adjuvanted vaccines, and then Vincent commented on the fact that they have different adjuvants. You might ask, well, how do we know it's not something in one of the adjuvants for one versus the other? But they mentioned that there was another vaccine that had that same adjuvant as the Pandemrix. The one in Canada is known as Arapanrix, and it is manufactured with different steps, which would result presumably in differences in the vaccine, the viral proteins, and so forth themselves. And so it could be that that's an explanation. Although they say, going on, signal of vaccine-associated narcolepsy was noted in Canada. Let's see, the 10 cases outside of Canada were linked to Pandemrix. In Canada, they were linked to the ones receiving the Arapanrix.
Yeah, right. Which they didn't look into.
Right.
They only compared the NP of these two, Pandemrix and Phocetria, so I don't know what.
Right. We don't know the level in the Arapanrix.
So there are 4 cases in Canada, Yeah,
I'm not sure the adjuvant is entirely off the hook here.
Right. So that was one point I wanted to make.
Yeah. So they point out that this MHC allele, HLA-DQB1 star 0602, which is linked to narcolepsy, in which we showed binds the variant peptide really well from NP. It's also been known for a while that it's associated with increased antibody against NP in infected and immunized people. So this allele hits this NP peptide particularly well. It binds to it particularly well. It's known to actually, this is a NP peptide that is known to be presented in this in MHC. They have an interesting speculation. So you can imagine in an infection with virus, this NP peptide could trigger autoimmunity. And they say, after 1918, the Spanish flu outbreak, there were episodes of what was called sleepy sickness or encephalitis lethargica. They say this could have explained it. And they say it could also explain the cyclical associations of narcolepsy with seasonal influenza infection in China, where they point out that vaccination rates are pretty low against influenza. Yeah. So, it could be.
Well, and there's an association, as they note in this paper, there's an apparent association with the H1N1 pandemic virus with narcolepsy in China as well. So it's not just the vaccine that can cause this. And, of course, it's possible that for these kids who got pandemics and then got narcolepsy, they might have, if they hadn't gotten the vaccine, they might have gotten the flu and then gotten narcolepsy or worse.
So if you don't get immunized and you get infection, you're at risk for narcolepsy. But, of course, even being immunized, the protection isn't 100%.
Right.
So you could still get infected and still develop narcolepsy.
I think on balance, it was still worthwhile for these folks to get immunized. I mean, the risk of narcolepsy from this vaccine was still very, very small. Obviously, it's something worth eliminating. But it was still, on balance, better to have gotten vaccinated than not to have gotten vaccinated, I think.
Right. They state that in the very last paragraph of their very long discussion section, in which they say, in individuals genetically predisposed to narcolepsy, exposure to influenza infection may potentially lead to stronger NP antibody cross-reactivity with the HCRT receptors than with the vaccine. So the benefits of the vaccine far outweigh the risks of complications from the vaccine.
So is there any therapeutic approach to narcolepsy based on this data?
There is already a treatment for it of some sorts. Yeah, they did.
Which does interfere with the interaction of antibody with the receptor or the epitope.
I don't think it has anything to do with HCRT, the treatment.
I think by the time you get the symptoms of narcolepsy, the autoimmune response against the hypothalamus is already done.
Yeah.
So
the clinician that I asked about it sent me something from Up to Date, an online clinical resource where they get treated with stimulant medications such as modafinil, methylphenidate, or amphetamines.
Mm-hmm.
Goodness.
Yeah, right. Yeah, somewhere in here that was. Another thing I wanted to point out is that so people can have antibodies against HCRT receptor, but they don't get narcolepsy. So it's not 100% penetrant. And they have this interesting discussion where there's a higher risk in kids, right, who got pandemics than adults. And they speculate that this has to do with a uneducated immune response in children compared to results. So in children, repeated infections may trigger autoimmunity because we haven't yet learned to make a very regulated and specific immune response, which is what happens later in adults. So it's not only genetic, but also has to do with what age you are as well. It's quite interesting.
Also, throughout, they look at this population that always puzzled me. These children from 2004 who didn't have narcolepsy, but they had antibodies to HCRT. And they showed that kind of like the pandemics vaccine in that binding assay, they had some pretty high results. And I can't remember whether the discussion resolved that sufficiently for me or not.
No, they didn't. There are, so it's not 100% exactly. And and they actually are very careful to say we really. These results are a step in understanding, but we need more confirmatory studies, different vaccines, different populations, more samples that are timed properly. Yeah, They admit that it's not perfect.
They have a lot of caveats in here. I wondered if they were asked by the reviewers to
Probably.
put them in.
They probably were, yeah.
And there was a line of text earlier, and in fact relating to the whole adjuvant thing that was a long parenthetical comment that was really out of place, and I thought, I think they stuck that in for the reviewers.
Stuck in at the end, yes.
Yeah. But, yeah.
I mean, I think the point is that it looks good that this NP peptide is involved in some kind of autoimmunity, but people have antibodies to this and they're fine, so it's not the whole story.
Mm-hmm.
Something else is going on. Mm-hmm. They do say, though, at the end, the benefits of flu vaccination currently far outweigh the risks of complications like narcolepsy and those related to natural infection.
And I think even without this being a lock on the NP peptide being the sole culprit or primary culprit, this does give enough information to vaccine manufacturers that they can look for this.
Yeah.
Mm-hmm.
And this is, whether this is the undisputed cause or not, this is clearly something you don't want in your vaccine. This is a particular sequence of NP, and you can now go look for it.
Yeah, you can imagine introducing a regulation in the manufacturer. The final product can have no more than X amount of NP or something like that.
Right.
You can also extend it to thinking about other vaccines and other HLA, very specific HLA types that might be associated with other things that would be a bad combination of patient and that particular vaccine.
You could potentially prevent this from happening in other vaccines where it might have been a possibility. Yeah.
So it might be a good idea to take all the sequences of the proteins that are in known viral vaccines, right, and compare them against human protein database to see if there's any more mimicry, right? Mm-hmm. I'm sure you would pick up something because there's a lot of...
Yeah, you'd have a lot of noise in that, but you might find some interesting leads.
So this is relevant to the rituximab paper we just talked about, because it could be that in some patients, maybe they have a particular HLA type that makes them make autoantibodies to a viral protein that's reacting with their own tissues. So I think it's worth looking in them and seeing what antibodies they do make. What are they reacting with? And where is the protein? Is it in a place that could... I mean, it seems likely that this could be a similar situation where you're making antibody to a neural protein, and that's giving rise to, in this case, narcolepsy. In the other case is chronic fatigue syndrome. So, interesting. Coincidentally, they are similar, but it wasn't planned.
No.
It was not planned. Did you like that, Dickson?
I loved it.
All right, this episode of TWiV is also brought to you by ICAC ICC 2015. ICAC is the well-known meeting of the ASM, International Interscience Conference on Antimicrobial Agents and Chemotherapy this year. In 2015, it's going to be combined with the International Society of Chemotherapy. We have a joint meeting this fall, and it's going to be a great meeting, over 110 sessions, 19 courses in drug development and infectious disease research. You can get continuing education over 30 hours. There'll be 6,000 people from all across the globe. There'll be exhibiting companies as well. And where is this going to be? Does anyone remember? Let's take a look. ICAC is in San Diego from September 17th through the 21st. So assuming California doesn't drop off the earth, this meeting will go on. This is September. So the registration, discounted registration rates end on August 6th. So if you want to, making a little correction there, Kathy.
Yes. Earth not flat.
So they're professionals from around the globe, not across the globe.
That's pretty funny. Well, this copy,
we're not on Discworld.
This copy was written by ASM, of course. ICAC.org/ICAC-ICC. Check it out if you're interested in attending. Now, let's do a few emails.
Oh, good.
And let's start with Kathy. That's Alice's there.
Okay, Alice writes, I want to share the source of Mice Lie and Monkeys Exaggerate with you. I was catching up with my thesis advisor, Dave Weiner, at University of Pennsylvania, and I asked him if he knew the origin of the phrase, mice lie and monkeys exaggerate. And he said, mice lie and monkeys exaggerate is a quote from myself. I gave a talk in South Africa, in Cape Town, on the use of non-human primates in HIV vaccine studies. And I went through animal models, and that was one of my conclusions. I don't believe it was the first time I used it, but it caught on there. I've used it for quite some time, and it's been quoted in Science. Paul Offit uses it in talks and has credited me with it. So for what that's worth, that's where it comes from.
Very cool.
Huh. What would be the likelihood that she would ask him and say, yeah, it was me?
Exactly.
Well, she probably had heard him say it, and
Right.
maybe she heard us say it and wonder where it came from.
Well, cool. It's neat.
Yeah.
I did Google it a lot, and I couldn't find out where it originated from, so that's a nice saying.
Well, now when people Google it, they'll find us.
And now we have...
Now we have the answer.
We have... what was it that Steen said? Mouse-eared cress does not lie.
That's right.
No.
Wow. All right. Thank you for that. I lost my place. Okay, we're at Wink. Wink writes, MERS coronavirus, like other coronaviruses, is thought to spread from an infected person's respiratory secretions, such as through coughing. CDC.gov. And this is probably from something that's very old, which we can't remember. But thank you. Let me read the next one. Edward writes, hello there. Edward from Alberta, Canada here. Just want to say that I've recently started listening to your podcast and am hooked. I'm just a simpleton, but I'm finding the show intriguing and educational. I'm finding myself listening to several episodes a day trying to catch up. Keep up the great work. You're not a simpleton.
No.
No.
Oh.
If you're listening to TWiV, you are by definition not a simpleton.
You are a complexilton.
There you go.
Alan, can you take the next one?
Sure. Stefan writes, hello everyone at TWiV. I'm an ambitious and subsequently naive 18-year-old who is insightful, unusually insightful for an 18-year-old, who is looking to get into the field of cellular biomechanics and microchemistry. I love your podcasts, and it's killer education, and it is amazing. The segment in 342 regarding aDNA and the acidothermophilic virus mentioned hydration was brought up as a primary cause of the formation of aDNA, as well as the sporullic qualities that may have caused both those viruses and the gram-positive sporulli, spores, to evolve the ultra-durable nature of the organism. My extrapolation would be to ask if there's any way for typical bDNA, say that of an antibody, to be forced into an aDNA structure and have those undefined proteins that were mentioned bound to the DNA. An application to an antibody would be far-fetched, most likely, but implications such as cancer cell developing those qualities would be terrifying. It may not be plausible for human tissue, but it could possibly be used as a time-release mechanism. HIV and other immune-related disorders. Thank you so much for looking at this, and sorry if it is nonsensical for the most part. I greatly look forward to continuing to listen to your show and learn in the field. Okay, so...
I'm not sure I understand the question.
I'm not sure I understand either. So you're talking about when you refer to the DNA of an antibody, I guess you mean the rearranged locus that encodes that antibody?
I suppose, right?
Mm-hmm.
But then he's talking about antibody binding.
Yeah, I'm not sure I follow.
So, is there any for typical DNA to be forced into an aDNA structure? Well, you put it at pH 2 and 90 degrees, and
yeah.
maybe that can happen.
That might do it.
I mean, that's the point about the phage, right? That's the idea that that those conditions plus the protein trigger it, and that's what happens in sporulation, right? But so yes, so in sporulation we have bDNA, and then under harsh conditions it gets converted to aDNA in the presence of a particular protein. So that's the idea, but I'm not sure I understand the antibody.
I think he's just
cancer.
wondering whether there would be some proteins that could force DNA into a different format, and if that DNA were DNA encoding antibodies, then that could have some repercussions.
Yeah,
that could be. Yeah, I think that biologically, the things like that are most often regulated by things like transcriptional enhancers and promoters and suppressors, and at least at the DNA level.
I don't think we know of such a protein.
Yeah, we don't.
In the case of this phage virus, since it's archaeal, the idea is that you need the protein together with the conditions, the environmental conditions, to cause the transition. So I think the protein alone is not enough. But now I get it. Right. Nice, Stefan. It's remarkably sophisticated.
Yes.
18-year-old.
Yes.
Well, keep on listening. Cool.
Indeed. Keep on writing.
Dixon.
Yep. Gunther or Gunter, depending on which spelling you go by, because it's listed in both directions here, writes,
It's my
TWiV,
my
is that you? Okay.
Yeah, I put the H in.
Okay, fine. TWiV is like a book without summary or without a table of contents or index, or like a paper without abstract. You can read through it sequentially, but it's hard to look up things, including links, switch forward and back, or skip chapters. By using MP3, you want to force us to listen to the whole thing or avoid it altogether? I think TWiV is too good to be lost to the MP3 archives without being able to Google for keywords later, or to quote from and to discuss in other forums or blogs. Good point.
So I replied to Gunther. You got any suggestions?
How would you like to archive?
How would you like to archive? Would you like to transcribe?
So
Right.
obviously a transcription would
or cross-index?
do it.
Yeah.
But that's a lot of work.
It's
Now,
a lot
I
of
understand that Google is working on a way to archive MP3s and make them searchable,
Oh.
right? So if they can figure that out, I don't know how that would work, but
Yeah, this is not a TWiV unique problem. There's
Yeah.
a huge quantity of human creative output and information that is now being encoded in formats we can't search in quantities that we've never had to deal with before.
I think
And since we've fired all the librarians, we don't have anybody to index it.
Yeah.
so. Since we've added timestamps, I think it helps. So you don't have
Yeah.
to listen to the whole
Right.
thing, right? But
Great if you had a voice recognition system. That would
Yeah, so he said he would. This fellow has written many times over the years and has made many comments on our blog, and he's a computer guy. And he said, "I'll look into this. Maybe there's some way to do this." But I know it's a universal issue that you can't search it at the moment. But
I'm sure.
someday,
Obviously, Google has a huge motivation since they own YouTube to
yeah.
to make this stuff
Oh, yeah.
searchable by what's being said.
Yeah.
That brings us to Eric, and you, Kathy, again.
Okay, Eric writes, Dear TWiVers, in TWiV 339, after the discussion on population growth, Vincent, possibly jokingly, commented that the problem will be no jobs. As a listener pick, I would recommend Economics in One Lesson by Henry Hazlitt. The lesson is given in the introduction. In economic policy, i.e. government rules, etc., one must examine all the effects, not just the ones that can be seen to benefit some, parentheses, usually the politicians benefit the most anyway. As an example, the push for a $15 minimum wage should rather be called the Robot Subsidy Act, since the effect of such a law will likely replace all jobs lower than $15 an hour in productivity with machines. The
Minimum wage.
Minimum wage, yes. I just was going to say molecular weight, and the minimum wage also could be called a minimum skills law, because its effect is to cut off all workers who have lower skills from getting to the lowest rungs of the employment ladder. After all, the minimum wage law does not require that anyone be hired, only that it is forbidden to hire at a lower rate. You can tell this is true by looking at who is pushing the Minimum Wage Act the most, higher-priced union workers, for one. As is often the case, the reason that some push for government act is to put a clamp on competition. A minimum wage law, of course, hurts the youngest the most, and because they become unemployable by government decree, they will need to find work in the black markets, most likely in drugs, prostitution, etc. So before you think that there would be no jobs in the future, remember that it's laws that restrict jobs, minimum wage, license, etc. And if you think that working for $5 an hour is bad, how about $0 an hour with no job at all? And that $5 would very likely go much further if the government would get out of the way. To plagiarize from another great show, another TWiGov is parasitic. Regards, Eric.
Thank you for the libertarian view, Eric.
Indeed.
Yeah.
Thank you.
Yeah.
What, Ron Paul?
We have had.
Although the point about the minimum wage law is valid in that hiking the minimum wage too high would inherently tend to cause unemployment.
I might have said it jokingly, but I do think if population growth increases, we will not have jobs for everyone. As it is, many jobs are in the service industry, which a lot of people are probably not happy with.
No, I don't think that's a problem, because as the population grows, the need for services grows.
Oh man, how many McDonald's do we need?
Well, how many people do you have to feed?
Starbucks. It's everywhere, on every corner.
And as the population grows, the businesses grow. You couldn't have had that many Starbucks outlets 50 years ago because you didn't have that many people shopping at them.
Yeah, well, it's also a matter of making people realize they want something that they never wanted before.
Right. I mean, that Starbucks may be a bad example, but you certainly need a certain number of supermarkets to sell the food that people are going to eat. You need to all the basic supplies of life are going to have to come through some economic process, which is going to require labor, and the amount of labor is going to be proportional to the population.
The problem is, if you don't pay them enough, then they will be unhappy and create crimes and deal drugs and all this stuff. Right?
There is some need to.
All right.
Yeah.
This is not TWiGov. Sorry, I'm very sorry, but we are human and we do worry.
They used to have a segment on Saturday Night Live about this, by the way, but it had some rude comments from each other, so we won't go there.
What was that called? That segment?
Jane, you ignorant.
Oh yeah, right.
Oh yes.
It's like ping pong, political ping pong. You've got these two interesting opposite views. Both have validities and non-validities. So interesting.
All right, let's make this last one. This is an email from Anthony who sends us a link to an article in the Chronicle of Higher Education called The Attack on Truth. We have entered an age of willful ignorance, and I recommend it highly to everyone. This is all about the concept of truth, and it's wonderful. Politicians pronounce that global warming is a hoax. Vaccinations, parents say we're not going to vaccinate. What is going on? And why are we willfully ignorant? And Kathy has highlighted a part here which blames the internet in part. And I like this. It facilitates not only the spread of truth, but also the proliferation of crackpots, ideologues, and those with an axe to grind. It's wonderful.
Right. And it lacks the editorial gatekeepers who can vet information. Outright lies can survive. Yeah. And go ahead.
They say, okay, maybe journalists could be the solution to the problem, but we now find that even mainstream journalists and news media are dangerously complicit in the follies of those who seek to disrespect truth. And then you pasted something at the end. Oh, yeah.
Frank Bruni in the New York Times on the 4th of July had an editorial entitled California, Camelot, and Vaccines. And he wrote, although the internet could be making all of us smarter, it makes many of us stupider because it's not just a magnet for the curious. It's a sinkhole for the gullible. It renders everyone an instant expert. You have a degree? Well, I did a Google search.
That's wonderful. Right. Yeah. It's a pity because it has great potential, right?
Yep.
Mm-hmm. Which is, illustrated in many great things, not just the Twixes, but all the other great stuff that's out there. But there's even more garbage and lies.
That's true.
And videotapes.
Conspiracy theories.
So I don't know.
If you read Yahoo,
what should we do about it?
Well, just keep reading.
The libertarian approach is to ban the internet, right?
No, you don't.
No, that would be the opposite of libertarian
That's right.
approach. The internet is almost inherently libertarian as it is.
That's
Theirs
right.
would be to remove all regulation.
Yeah, they don't want to regulate anything. I'm sorry.
Right.
We've been having a libertarian arc on TWiM about making restaurant workers wash their hands. And a fellow's been writing, and he said, I'm a libertarian, and I don't think you should make restaurant workers wash their hands. If the restaurant spreads infections, it'll get shut down.
You know what's missing is a dialogue. I mean, when you go to the internet, you read something, there's no one there to say, oh, and also you should read this for the opposite view. There's no intercourse from intellectual exchange. And so my recommendation is stay in school because you get those exchanges in classes. You don't get them when you're one-on-one with the internet. You either trust it or you don't, and that's the big difference. And to find people that are just gullible enough to believe whatever they've read, and it supports their own theories, of course, then they have their arguments all lined up. And like Alan says, you can't argue with the creationists. They've got they've got their literature, and their literature says this, and you're not going to go beyond that. So, but staying in school allows you to hear all of the views, at least in most schools. Not all of them, of course, but in a lot of them it does. And that's where that's the validity of it, is that it creates a discussion.
Well, I guess what we're
discussing.
what we're discovering is that when you fire all the big bad gatekeepers like the mainstream media, weird things happen.
Weird things happen.
Let's do one more. I just found one I'd like to read. Alan, can you read Christina's? I just pasted in there.
Oh, now I've got to scroll back up again.
Yeah, sorry about that.
End of TWiV email.
Just
Then I've got
that, it should be one from Christina.
Christina. Hi, Dr. Racaniello. I'm Christina, a Filipino PhD student at the University of Georgia. We met when you visited UGA last spring. I wanted to follow up on a letter that John wrote on TWiV 342 and ask for her or his email. Maybe I could give them some tips as well as some contacts. I totally understand John's frustrations because I was in the same position as they were a few years ago. Luckily, I have a supportive mentor, faculty, family, and friends that made it possible for me to pursue my dream of becoming a virologist. Thank you so much for TWiV, not only for making virology accessible to a wide audience, but you've made it possible for people to connect. More power to you and the Twix team. And by the way, it's 31 degrees C and 35% humidity here in Athens, Georgia.
31 degrees, that's all.
That's warm.
All right, so I did connect. I think that's great. I did connect Gian and Christina, so thank you.
Excellent.
And then just one more, I'll do this one. Suzanne writes that basically, it's also with respect to Gian, she suggests to crowdfund.
You mean like Greece?
Application fees and travel. So she says, this time around I heard you reading John's letter and I had the thought that if money for application fees and travel turned out to be the main hurdle for him to get a good shot at a virology program, maybe he could crowdfund it. I bet there are listeners who'd be willing to chip in. I would. Thanks again for your podcast, and as well as all the fascinating info and discussion. Thanks for introducing us to the music of Ronald Jenkins. I've since bought all his albums.
Oh, wow.
They're great. And we use Ronald Jenkins music on TWiP and TWiM. RonaldJenkins.com. So I wrote an email to Ronald Jenkins. I passed on Suzanne's, and he was very excited. Ronald's a cool guy. He said, thank you. Thank you so much. All right, so let's do some picks now.
Excellent.
Alan, what do you have?
I have. This is an article from a home brewing website that I stumbled across a little while ago. And it is a very in-depth analysis on the science of beer aging. So if you're into beer,
I mean, drinking it, but not aging it.
Oh, okay. Well, there are certain beers that are good to age,
Really?
like wines, and for some similar reasons. And so this discusses the esters and the various other chemical compounds and why particular types of beers age better than others. And I found it really cool that somebody has actually laid this out in a somewhat approachable fashion. I mean, there's some chemistry and there's some structures in there, but I think it's fairly well explained.
Do you make beer?
I haven't for a number of years, but I did in graduate school, and I mostly buy beer, but I do enjoy the stuff.
I didn't know that some beers were aged. I thought
no
you
idea.
should
I
drink
didn't
beer
know.
as quickly as it's made.
No, I at my thesis defense party, I served a barley wine that I had brewed, which I had brewed the
Really?
previous year, and I aged it for a year, and then turned out very nicely.
I don't remember. Was I
Well,
there?
after if you had more than one bottle of it, you probably would.
Did you make a custom label for your defense?
I did. Did I do a label on that one? I think it was in Grolsch bottles, and the labels wouldn't stick. But
Right. Yeah, Grolsch, that's true.
Those are good bottles.
I have a pick which really is for Kathy. Caught my eye. It's an article over at Fast Company. Why Lego is spending millions to ditch oil-based plastic. And it's really cool that they want to replace by 2030. Right now, their Legos are made from ABS, which is an oil-based plastic. They want to replace it with a non-oil-based thing, and they make 60 billion blocks a year.
Good Lord.
That's a lot of blocks.
Good heavens.
And they're putting a lot of money into doing this, and that's cool because you should get away from oil.
I actually found this figure kind of stunning. 3/4 of Lego's carbon footprint comes from the extraction and refinement of oil used in their toys,
Holy cow.
Yes, that's a
which
big deal.
suggests that their factories are actually really energy efficient.
Yes.
They did say that they recycle their own plastic when they can, but using plastic from the outside, they can't be sure of what else is in it.
Right. Recycling plastic is very hard.
Yeah.
Anyway, I thought you would like that, Kathy, and everyone
Yeah.
else as well. What do you have for us, Kathy?
I picked a link to the University of Michigan College of Literature, Science, and the Arts webpage where they have the winners from the Art of Science contest that they had. This is the second year of it. You can also find... Oh, no, the link didn't work to the previous year's one. But anyway, art done by, it seems like mostly undergraduates, not necessarily all science majors. There's an art and design, architecture major. There's a engineer. And I just thought there were some cool renderings of things, scientific, that are artistic.
It's
I like
nice.
the mosquito with the capillaries and everything. That's very
Yeah,
cool.
yeah.
Yeah, that's
So-
neat.
That's amazing.
Very cool.
Yeah, I don't know what. I guess maybe that one was made with a 3D printer, or the parts of
Wow.
it were.
Wow.
Maybe.
Yeah, malaria parasite. Yeah.
Love
Oh,
it.
that one. Yeah, that one is 3D printed.
Love it.
Okay. Good.
Or digital 3D model. Sorry, so
Oh.
that's a digital image.
Oh.
It's still beautifully done.
Yeah. So check it out.
Cool. Dixon, what do you have?
Well, it's a last-minute addition to the list, but I've had this article sitting around for a week now, and it's from Science Magazine because I'm a big fan of cloud form, the process of cloud formation. Okay, because you can pin it on a single organism, Emiliana huxleyi, which is a
yeah.
coccolithic phytoplankton, by the way.
And there's a
So-
virus of it too, you know.
And there's a virus of that too. So the title of this article in Science is called, it's from the Marine Sulfur Cycle section, which is their only one article. It's called Identification of the Algal Dimethyl Sulfide Releasing Enzyme, a missing link in the marine sulfur cycle. And they immediately in their abstract acknowledge the fact that the algal blooms, which are important for food webs, also secrete dimethyl sulfide into the atmosphere where it can affect cloud formation. And in fact, a lot of marine geologists believe that that is the dominant form of mechanism for making clouds. And so I thought it was fascinating to see some of these biochemical analyses of this organism finally coming to the forefront, because I think the more we realize that this is a precious organism and the oceans are being so impacted by environmental changes, what if we couldn't form clouds as a result? This would have a devastating effect on the terrestrial biosystems that we are all part of. So I picked this article to draw attention to the fact that we are all linked together in some interesting web of life that begins with the DNA molecule.
So this thing, the Emiliana makes these huge blooms.
They
right,
do,
in
and they
the
sequester
oceans.
carbon too, because their outer shells are made out of calcium carbonate. So when they form these large aggregates and they release their dimethyl sulfide, the entire colony sinks down to the bottom of the ocean and recycles carbon as well as volatilizes sulfur and puts it in the atmosphere.
And
Now, they have a major impact on cloud formation, but clouds form for physical reasons.
right.
Well, Alan, that's true, but they can't start unless they have dimethyl sulfide as a beginning product.
So there were no clouds before life?
That's probably maybe there was just an intense atmospheric humidity. But yeah, that's one of the conclusions that you might want to draw from this. You should. I'll send you some literature on it if you're interested, because the ultimate fate of the sulfur part of this is to form sulfur dioxide and then sulfate.
yeah.
And the sulfate then is a particle. It's not a gas or a liquid. It's in a solid, and it gloms onto particles in the atmosphere.
So that forms the nucleating centers for the
That's
droplets in the cloud.
Correct, and it's hydrated, and it forms sulfuric acid, which is a hygroscopic chemical, which then attracts more vapor to it, and in fact, it forms enough to make a cloud. Before that, you can't make a cloud.
Huh.
And
So it's quite fascinating.
What's cool is the viruses of Emiliana control the blooms, and they regulate cloud formation.
Right.
So then, when it rains, the sulfur returns to the earth and begins the cycle again.
Yeah.
So that's a fascinating. I love it. I just love it.
And on TWiV 34, we talked about how the viruses of Emiliana
There you go.
regulate the population. So check
about
that
that?
out.
So that was my pick.
Thank you, Dickson.
You're quite welcome.
We have a listener pick from Steven. Last week, The Martian trailer was picked, and it was mentioned that The Martian could get a record-breaking 3 picks of the week: the book, the trailer, and the movie. Let's push that up to a potential 4. I'd like to pick the audiobook version of The Martian with excellent narration by R.C. Bray. Has won an Audie Award in its own right. Highly recommended.
Huh.
Wow, 4 picks of the week.
Wow.
Unbelievable.
How about that?
From something he wrote just,
Huh.
out of his interest in space, and he gave it away for free for a long time.
Wow.
It's great.
Wow.
Good for him. That'll do it for TWiV 3, 4, 5. You can find it at iTunes and also at TWiV.tv. And if you have a mobile device, a cell phone, a tablet, and they run an iOS or an Android operating system, you can get a podcatcher and listen to TWiV as well for free. These podcatchers are often free, but they subscribe to podcasts, so check it out. And do send us your questions and comments to TWiV at TWiV.tv. Dickson Despommier can be found at verticalfarm.com. Thank you, Dickson.
You're welcome.
Have a great trip.
Thank you. I might download that book and take it with us. Audiobook.
Yeah, you should read it. It's
Martian.
really good.
I think
Yeah, my brother listened to it as an audiobook, and he liked it that way
I'm going
too.
to do that. I think I'm going to do that.
Yeah, it's a great book.
Yeah.
And last week we had the trailer. It was fabulous. Can't
about that?
wait.
Huh.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun.
Alan Dove, AD, can be found at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. Hey, I got to tell you guys something. I got the coolest thing. I got a vanity license plate.
Oh, come on.
It says viruses.
Really?
All right.
You got it?
Awesome. That was available?
I could
got
not
it.
believe it.
Outstanding.
Last week I said, I want this license plate. I don't know why. And I probably had something to do and I didn't want to do it. So I went and got and they said, we have confirmed your order. It should be shipping
my
shortly.
God.
Viruses. Can you believe it? I'm so
Yes.
excited.
Yes.
I'm very excited.
Outstanding.
We definitely need a picture.
For sure.
Oh, yeah.
I mean, I'm just a child, but I love my viruses.
And they love you.
So now you'll be driving the TWiVmobile.
That's it.
I was thinking of TWiV, but, I figured viruses, everybody knows
Viruses,
what
that's
viruses
better,
are.
yeah.
Virus was taken. I tried that first.
Okay.
And then I tried viruses, and I was just amazed.
How about that?
Thank you, Governor Christie.
Oh, no. Oh,
No, he has nothing to do with it, right?
That's right. That's for sure.
Did you notice, Dickson, they closed the lane of the GW Bridge this morning?
Another time.
damn it.
By the way, there was a national public radio broadcast this morning which advertised the fact that of all the states that manage their budgets, New York State is ranked 47th in the nation, and New Jersey is ranked 51st. There's only one that's worse than ours.
Which
Wait,
one-
49th. Sorry.
Yeah, I was getting confused there.
No, there's the Philippines, and then there's
Are we including Puerto Rico and
no. Okay,
Colombia?
we're next to last. We are next.
I'm not surprised.
To last.
And he wants to be president.
Don't
yeah.
tell me. Mississippi?
Right.
Oh!
How would you know that, Alan?
Well,
You can always count on Mississippi.
that's right.
Oh, wow.
They come through in a college.
So for quality of roads right now, it's Michigan.
Really?
And amount spent per capita, I think,
You're
is
the-
also we're 50th on roads.
Oh, you're
Gee
50th on roads?
whiz.
You have bad roads?
Oh, yeah.
Really?
Really?
I didn't know that.
The hub of the Motor City.
Yeah.
And by the way, my kinfolk are mostly from Mississippi.
Well, I'm just down the road from New Orleans, so.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.